Hey, and welcome back to the show. I know it's been a while since our last episode, but we're pretty excited to be back. Feels good to be back in the chair. Today we'll be looking at our third and final paper in the CRISPR series. But this one's a little bit unique because instead of being a biological application, this is actually a software application or hardware application. So it relates to microelectronics and all that stuff. So that's pretty cool. The name of the paper is a CRISPR-Cas-inspired mechanism for detecting hardware Trojans in Fpga devices. The researchers working on this paper were Dylan Staub and Rashmi Jha from the University of Cincinnati, and David Kapp from the Air Force Research Laboratory.
I'm Hunter.
And I'm Luke, and you're listening to EpiSTEM, a show where we explore papers in STEM fields and let you know what they're all about.
I'll talk a little bit about the abstract of this paper. So, nowadays, because there's a lot of hardware and software implementation in almost any industry, the semiconductor field is really globalizing, at least the supply chain of it. With that adds a lot of risk to have viruses and or other errors occurring, whether it be malicious means or someone just messing up in general. So, this paper will go over the Trojan detection in Fpga, which are field programmable gate arrays, which we'll explain a little later. And currently there are three largely used methods, and those are test time implementation, pre-implementation, and runtime methods. The researchers in this case believe that they can utilize methods similar to biological means such as CRISPR to detect Trojan viruses within a system. If you've been listening to the CRISPR series, the basis of this is essentially to find the bad viruses in the case of Trojan hardware viruses and remove them in a similar splicing method as CRISPR does in the human body and other biological means.
Now might be a good time to talk about Fpga applications.
Well, FPGAs, field programmable gate arrays, they eliminate the need for heavy processing unit times within a system and make the system simpler. So in essence, let's say you set up a system that has a switch, a light, and another switch. With a regular system, you would have to use a processing unit to process the commands or the code given to the system and then tell the switch to flick on or off, and then the light would then correspondingly turn on or off. Same with the other switch. However, if you're using an FPGA, you're able to just have an input, whether it be a voltage or some other means of input into the system, and then the FPGA will just do the action that you want it to do, such as a switch turning on or off, and then subsequently turning the light on or off. So a good example of this in a non-technical way is having one person do 10 tasks at once. It's going to be very time consuming, and they're going to have to spend a lot of time on one task and then the next one, and so on and so forth. And like that's the processing unit. So when a processing unit has a lot of tasks, it doesn't do them very efficiently at all. Whereas if we have an FPGA, that's like having 10 people ready to do the tasks, and then their manager tells them do the task, and they can all complete all the tasks simultaneously, so that everything is more efficient and gets done in a good manner.
So CPUs, while in that particular scenario, probably wouldn't be the best application or the best piece of tech to use. They're just one of many ways to control a circuit. Besides CPUs, there are FPGAs, which is the topic of this paper. But similar to FPGAs are these things called ASICs, which is ASIC, which stands for Application Specific Integrated Circuits. So an ASIC is basically just a circuit that's designed to do one particular thing on one particular circuit board to perform one particular task over and over and over again. Those are really fast and they're cheap, but they're only good for that one thing they're designed for. An FPGA is advantageous over that because it can be reprogrammed down the line to do other things that are maybe similar or other useful tasks in that circuit.
So because these systems are simplistic in nature and try to make things more efficient, that allows for people that want to make malicious hardware attacks or malicious software attacks to them. It gives them great ease to do that because these systems don't have that much complexity to them. They're more or less just circuit boards that are being given inputs and then they give out outputs. That's in essence what they are. So because of that, hardware Trojans, which are sort of hidden viruses per se, or hidden malicious hardware deformities, occur, and they're whenever someone wants to put that into the hardware itself, whether it be the integrated circuit board or into the software itself, which usually doesn't happen, it's mostly hardware issues. They're alterations to the circuitry that can be harmful to any of the processes that are occurring within the system. They can be prominent nowadays due to the Fpga globalization efforts, and due to the outsourcing of processing Fpga units, there is less oversight over the unit's hardware being made. So that takes away from a lot of the hardware design being able to be in control of the system and the hardware itself when it's being printed. So a lot of companies nowadays are utilizing a method called split manufacturing. So split manufacturing involves integrating circuits into multiple companies so that they're, let's say, half of their circuit board is printed or made in one company and the other half is used in the other company. So then not one vendor is utilized for the whole project, because then they could be implementing hardware Trojans into the system.
So in this situation, a hardware Trojan is just is it the same as a manufacturing defect? Is it a mistake? Does a Trojan happen by mistake, or is it somebody wants to put a virus on the device, or put some kind of like a surveillance device into the system or something like that? Is that what a Trojan is, or is it just a manufacturing oversight?
Hardware Trojans involve a malicious intent and the want of someone to actually mess with the programming and the hardware of the system, whereas an error or a defect would be considered something else.
So this is an issue that arises because with electronics growing and becoming more popular and more prevalent, manufacturing is being spread out and distributed amongst multiple companies, multiple entities, and that's just a harder thing to control. It's easy to manage quality within one company, but to try and manage the quality of the 15 companies that are making parts for you is probably pretty challenging. And that's what you're saying. Like, there's a lot more opportunity for someone with malicious intent to slide something in there.
Yeah. So there's the pros and the cons with split manufacturing here. Is that split manufacturing allows for you to split up your vendors and allows for you to have multiple companies input into the hardware, so then not one company has pretty much your whole property or your IP, like your intellectual property here. In this case, it allows for two companies to have errors or conflicting views on it. So there's a lot of pros and cons to it. Most people think that it's more pros towards split manufacturing, although it's at a higher cost. It just protects them and gives them more security, so that not one company is in charge of everything, or not one vendor is in charge of everything.
That makes more sense, because the benefits of splitting up your intellectual property, you don't want to give one vendor the blueprint to your whole product, so you give them just a tiny little piece of the pie. And that's probably a lot safer, because a vendor probably has less motivation to put a Trojan in if they don't have much to gain out of it. When if they got caught, they could lose your business. So it's probably more pros than cons.
And it's not about the trust within the vendors that's the problem. It's just that if someone that does have malicious intent is able to hack their way in or get the info from a vendor, they don't have all of the info that's sent there is the main issue. Yep.
More links in the chain, more possible vulnerabilities.
Exactly. And with that, there's a lot of different ways that Trojan insertion can be done. So it can be done during the fabrication of the device, which is the main method talked about within the paper. And there's a couple other methods, whether it be someone messing with the software, which is harder to do because there's a lot of control over the software internally, and that's never really outsourced. But there's also just, as you were saying earlier, defects. If defects are put to just a very high rate, such as quality control is not really doing their job and they're not taking charge of actually stopping a defect from occurring that could lead to an unintentional Trojan. which wouldn't be totally malicious intent, but it
Right.
could be detrimental to the processing of the unit. So with Trojan prevention, it's sometimes very time consuming and costly to do because that involves engineers and other people looking into the processing of the unit, looking into the manufacturing processing, the software processing, everything about the unit. So that gets very tedious for people and companies to set up and do. There's also Trojan detection methods, which can be complicated due to the volume of searching that needs to be done because they need to search through a lot of the systems themselves because they'll have different kinds of detection methods, whether it be destructive or non-destructive testing. So destructive testing would involve using a microscope-based test that would be looking at the circuit boards themselves and looking at, or I shouldn't say circuit boards, the circuit systems themselves and looking for any issues, but those don't pick up any of the software issues. That's all just hardware, which is probably it is the main issues here, but you also are just not worrying about half of the system, which is detrimental if you're looking for an error. Non-destructive testing in the Trojan detection methods would involve going through the code and having net lists found by IP vendors. In this case, that is Internet Protocol vendors. But this is very, very time consuming, and it's not always fruitful because sometimes you're going to be finding an error in the code that will help you, but sometimes you can just overlook it because this involves software engineers looking through lines and lines of code over and over for hours a day. It's very costly and can easily lead to someone overlooking something even though it is an error.
So just trying to find these possible Trojans is quite an undertaking, just because it's either super expensive and time consuming or not very effective.
Exactly. And so that's why a lot of companies nowadays are looking for new methods to find, to have a tolerance on Trojans and the removal of them. So like I talked about earlier, there are three main methods of Trojans being used within a system. So runtime monitoring helps to find Trojans within a system because it looks for redundancy in a system. Many systems that are using runtime monitoring utilize the triple modular redundancy method, which is the TMR method. This uses identical versions of the IP that are redundant from different vendors. This works well under the assumption that Trojan does not exist in all three versions. So in, I guess, some simpler terms, it is pretty much they're getting three different vendors to send them the same thing with the same IP, which usually if there's something that's occurring such as a Trojan or another error, they would have a differing IP. So the redundancy actually helps them to find if there's an error or not. It's like an overqualification, I guess.
Okay.
Yeah, with current methods of CRISPR implementation in digital systems, Trojan detection or prevention is nearly impossible due to cell models being incomplete and very complex, as stated in the paper. In 2012, J. Craig Venter Institute research reported a basic whole cell simulation of Mycoplasma genitalium, which accounted for all the pathogen's molecular components and their interactions. So, with this simulation that they made, it was very in depth, but that organism only has 525 genes, which is very small for what you're trying to look at. So there's, in comparison, E. coli, which is a very common organism that people are looking at, has 4,288 genes, and human cells are way more complex to that. So there's a lot more that needs to be done, because that simulation that they did took 10 hours to generate, and it generated 0.5 gigabyte of data. So that is a very time-consuming and very data-consuming process for doing a simulation at even a small scale. And later on, people found out that the simulation wasn't even perfected. There were some minute errors that were occurring within the system, and they later fixed them, but they could not totally adjust the processing times that they were dealing with.
And that's kind of like the nature of a Trojan virus in general. It's why they give it the name that it has, right? It's that you can't really detect it until it's too late.
Exactly,
It's
just like the Trojan horse.
Truly.
And just like the people of Troy should have done, the researchers are setting out to make a realistic CRISPR process model using Python to eventually help protect FPGAs from hardware Trojans. They were not concerned about the formation and movement of macromolecules. The main concern of their programming was how genetic information flows through a system and affects it. So mainly they were trying to figure out the process flow of everything and how CRISPR works within the organic systems, then they can bring it into a circuit system. They worked through the following principles in order: production and degradation of cells, cell diffusion, binding using CRISPR, DNA and RNA changes due to CRISPR, and PAM sequences, which are protospacer adjacent motifs. So you probably would have heard about the other parts of that in the rest of our CRISPR series. However, the PAM sequences were not mentioned in the rest of the series, and those are essentially the DNA sequence that follows the DNA target. So when people are using CRISPR to splice out a certain area, it's the sequence that follows that area or location. In the case of this paper, we might refer to it as the locus, but it's synonymous with location.
What I find really amazing about this whole idea is that they're effectively making a software/hardware system that like closely mimics something that you see in biology. It's a really interesting application of biomimicry. Usually when you see biomimicry, it's things like trying to make rope that behaves like a spider web, or trying to make gloves that behave like lizard hands. It makes some sense, but this is a pretty crazy application of biomimicry that we will talk more about in our Curiosities episode, which will be released next week.
Yeah, and the Curiosity episode will also go over a book that Luke and I have both read and found very interesting, called Biomimicry: Innovation Inspired by Nature by Janine Benes. She wrote this book in, I believe, 1998, and it's still a very applicable book to any engineering or just STEM field in general, because so many things around us are based around biomimicking the nature of any other animal or other beings on the planet. And we will definitely talk about this more in the Curiosity episode of CRISPR, so stay tuned.
But back to the paper, the simulation that the researchers were using to test this theory is an example of biomimicry of the CRISPR process.
The simulation done by the researchers actually worked really well. They got what they were looking for. They weren't perfect, but they got there. They found that the system could be mostly simulated, and thus it could lead to using a similar method in FPGAs. One big difference between the CRISPR method and the simulation they used is that spacers are added less frequently with the CRISPR method compared to the simulation.
The spacers are complementary halves to a specific strand of DNA, and they're how an entity can remember that specific genetic sequence. The simulation simply has less of these built in.
This is due to the CRISPR method being very, very precise and being genetically pretty much modified over millions of years. Like, it knows what it's doing through the process, whereas this is a very artificial system that they set up here, just following the code. The results also show that more research is needed to find the percent similarity required for cRNA to target something. In the simulation, they use a 90% similarity rate, but they are not sure, and I don't believe that anyone that I know of is sure of the real similarity rate that CRISPR is utilized in organic systems. Through the other methods that we've talked about in our previous CRISPR episodes, they have been doing higher probability rates also due to it being an artificial process. But when they do a more natural process within the body itself, there is not data that I'm aware of as to the actual similarity rate.
Now, when you're talking about similarity rate, similarity to like a purely organic?
It's the similarity rate of when a CRISPR method is used, it's looking to splice out a specific gene sequence or a specific sequence of code.
How similar is the sequence to the target sequence?
Exactly. How similar the target is to what you're getting at.
I see what you mean.
So, Another thing that could not be simulated well is that cells carry out many functions in parallel, while a system such as the code is only able to do a few tasks in parallel. It's not able to do everything at once, which is the benefit to FPGAs, is that they're able to do more things at once than a regular processing unit or a CPU would be able to do.
Let's go ahead and take a quick break. So just before the break, we were talking a little bit about the simulation and how it went, and now we're going to talk a bit about the 4 phases of designing an FPGA.
There are 4 main phases that the researchers discussed for creating FPGA systems. So they go as follows: behavioral level realization, RTL realization, which is register transfer level, gate level realization, and bitstream realization. Now I'll go a little more in depth into what those actually mean. So behavioral level realization is when the system is very algorithm based. This is like the raw form of where the system starts. Because it's the root of the process, it's very primitive, and it's hard to implement any type of Trojan detection or prevention methods here. So they didn't even look into that very much at all. The next phase here is the RTL realization. As I said before, register transfer level. This is when aspects of the circuit design are created for the transfer of different registers in the system. Parts of this phase could be utilized for Trojan protection by implementing HPC, otherwise known as a hardware property checker, which would essentially check IC properties while the system is running. Earlier in the podcast, we talked about runtime errors and runtime detection for Trojan hardware, this is an example of that. This is a good possibility to catch Trojans under the pretense that the Trojan will cause a change in the system properties. So if it's not causing any changes within the system or causing changes to the system properties, this is not a viable option here. Studies need to be done on what triggers Trojans and their payloads so that there is a better idea of their signatures. As of now, the researchers of this article are working on getting more data on this and be able to further their research into this topic.
So what you're talking about here with detecting a Trojan in runtime, is that the right way to say that?
That is the right way to say that.
The issue with it is that you're only finding it after it's already affected the system. Like it's already possibly negatively affected the system, and ideally you'd be able to stop it before that, right?
Not necessarily affecting the system already. So with a runtime detection, you're able to detect when there's a process change. So whether that process change is malicious or not depends on the Trojan itself. However, if you are able to detect it early on in the process and be able to find that difference in the process, whether it be just a little change in the code, a little bit change in the hardware, you would be able to detect it while it's running and stop the process or eliminate and, as with CRISPR, splice out the bad data that you're trying to get rid of. And that's actually the point of having the security labels and the security tags, is being able to say, hey, that guy over there, we need to take that code, we need to take that guy out of here because they're causing some harm to the system.
Right, like that piece of code wasn't originally here. This is not an intentional piece of the code or not an intentional piece of the system. This is
correct.
This came to the party late. Like,
yep. They weren't invited. They came in and they gotta go.
In other words, who do they know here?
So the third phase of FPGA production and processing is the gate level realization. This is when logical links are set. For those that don't know about logic-based systems, these include things such as and, not, exclusive, or. They're pretty much determining factors to decide how the process is going to generate code and how it's going to process the code. So, Luke, you know a little bit about this,
and
yeah,
whatnot.
right?
Yeah, they're all just like really simple mechanisms for handling input. So, like an example, you mentioned an AND gate. That's just a simple like logical gate that given two inputs, if both of them are, like, it's all ones and zeros. So if it gets two ones, if both the signals are active, you would say, or if they're both live, or however you want to call it, then it would output a 1. But if neither of them was, if they were both zeros, or if one was a 0 and one was a 1, then it would output a 0. So that's an example of like a logic gate, just a real simple mechanism for inputs to follow.
Yep, exactly. So one way to implement Trojan protection here would involve glyphs, GLIFT, which is gate level information flow tracking. So this would put security tags or labels on the data as it's being processed so that the information flow can be tracked throughout the system. Thus, you can track the system if there's a malicious hardware or malicious software occurring. You can track where the process is pushing that information to, or where it is just in the process in general. The fourth and final phase of the FPGA production and processing is Bitstream Realization. This is the last stage prior to actual programming of the system, and it will be implemented really right before they send off to the vendors to get everything set up with the printing of the circuit boards and whatnot. The Trojan protection method for this phase would be similar to the gate level that I just described, and that would be involving information and process tracking. As of now, the researchers believe the Bitstream Realization is the most viable option and the best option for detecting Trojans, and it is a little costly right now due to the time and effort needed to put into this, but because it would be able to save the company a lot of money and be more efficient in general if they're able to detect these Trojans and able to detect any errors that they're going to be involving. So the researchers here believe this is the future of where we need to go with Trojan detection. Well, that brings us to the end of the beefy part of this episode. In conclusion, I'll give you a little synopsis of what the researchers did and how they did it. So the researchers worked to model the CRISPR process through Python coding, and then eventually talked about reaching out and using the same methods of CRISPR to protect FPGAs from Trojan hardware. With a successful trial, they showcased areas of the FPGA processing that could be utilized for Trojan detection in hardware. They found that bitstream level protection methods are the best ways to detect Trojans. With this, they want to work in the future to generate Trojan signatures on a bitstream level. They will be continuing research with themselves and other researchers to actually find a way to detect hardware Trojans in the FPGA processing and the FPGA generation. This brings us to our nerd notes for this paper here. So I just want to go a little more in depth into the breakdown of the simulation they made. I found it very fascinating. It's a very complex system, but I think they did a good job at actually pretty much coding the CRISPR method in Python, which is an immense task. I'll give them credit for that for sure. But here we go. So they started off by finding the CRISPR location and find the initial spacer that they add. The foreign DNA sequence is then stored for later use in the program. So this would be stored in the memory of the code. More spacers are then inserted into the location that they used from subsequences of the foreign DNA sequence stored earlier. So they take the foreign DNA sequence, put some little parts of it into the location that they're at, and then continue on from there. Once all the spacers are added, the same sequences are transcribed into crRNA. So to do the coding of this, they actually set it up so that the RNA per se is actually just the code telling the rest of the code to grab the foreign DNA sequence that was generated earlier, and then pretty much do a comparison. From there, they were able to splice the unwanted code or the unused DNA sequence, and then insert the foreign DNA sequence that they wanted to put in there. In essence, that's really what they did. Like I said before, it is a lot more complex than that. If you want to check it out, read the paper. It's actually probably one of my favorite papers we've done so far. Really enjoyed it, and we'll put in the link to it in here, and
Yeah.
Yep.
All right, so that's the end of the episode today. Thanks for listening. If you haven't already checked us out on Instagram, our Instagram is EpiSTEM Podcast. You can find any other episodes on any streaming service that you get your podcasts from. If you have any feedback for the show, you can email us at epistem.pod@gmail.com. Or if you really like the show, feel free to support us on Patreon. Our Patreon page is www.patreon.com/epistem. Again, today's paper was titled A CRISPR-Cas inspired mechanism for detecting hardware Trojans in Fpga devices. The research was performed by Dylan Staub, Rashmi Jha, and David Kapp, and supported by the Air Force Research Laboratory, and published by Cornell University's Archive Computer Science Journal. Thanks for listening, and we'll hear you in the next one.